Okay, now it's recording.
That's weird.
Okay, all right. Sorry about that. Okay, so hi everyone. My name is Kevin Flores. I'm a third-year entomology undergraduate in Dr. Quinn McFrederick's lab. And today I'll be giving a presentation on the phylogenetic analyses of a pectate lyase, HAMP domain-containing histidine kinase, and response regulator transcription factor gene found in the Apelactobacillus mitchneri clade. So for some background and significance, so microbes have been found on the pollen provisions of bees and in the guts of developing bees, and multiple studies have also shown that these microbiomes have strong impacts on bee health. Unfortunately, though, most bee microbiome studies uh, have focused solely on honeybee microbiomes and leaving wild bees, such as bumblebees and solitary bees, kind of in the dark. And so because of this, very little is known about wild bee microbiomes. And this is what our lab aims to address. And so in my project, I've done this by identifying quorum sensing genes in the Apelactobacillus mitchneri clade, which consists of these three lactic acid bacteria, Apelactobacillus mitchneri, Timberlakei, and Canuii, which are just lactic acid bacteria commonly found on like flowering plants. And among these clade, I found the quorum sensing genes pectate lyase and histidine kinase and response regulator transcription factor gene, which are part of like a two-component system. And so the purpose of my research was to make gene genealogies to explore the evolutionary history of these three genes to gain a better understanding of them. Particularly, I wanted to see how these genes were possibly inherited, whether through vertical or horizontal transfer, see their origin, gain an understanding of what possible functions these genes might have, and to see if these genes are unique to the Apelactobacillus mitchneri clade, or if these genes are found in any other lactobacilli. And so I did this by first searching for amino acid sequences of these genes by looking up their accession numbers that the program KEGG assigned to them. And I did this on NCBI. And so on NCBI, I found these three genes and I carried out blast searches to gain the amino acid sequences. So for pectate lyase, I did a blast search with max target sequences at 250, and I did this twice, one for a 369 amino acid sequence and another of a 412 amino acid sequence. And I required these sequences from the Apelactobacillus mitchneri genome that's available on NCBI. And then I used any of the sequences that were at or above a max score of 320, a coverage of 89%, and a sequence identity of 41%. For histidine kinase, I did the same thing, blast search, but this time with a max target at 500. And then I used any sequences that were at or above a max score of 429 and a sequence identity of 41%. And again, I acquired these sequences from the Apelactobacillus mitchneri genome for all the genes. And for the response regulator transcription factor, I did a blast search with the max target sequences at 500, and I used all 500 sequences that came up. Next, I aligned the amino acid sequences using the program Mesquite, and I then performed maximum likelihood phylogenetic analyses and visualized the resulting trees using Mesquite. So here we have the pectate lyase gene tree, specifically the 369 amino acid sequence one. And we can see that the gene tree has evolutionary relationships similar to that of the species tree of lactobacilli. So can you guys see my pointer by any chance? Okay, cool. So over here, we have the Lactobacillus mitchneri clade. Right here, Apelactobacillus mitchneri, Timberlakei, and Canuii. And these are pectate lyase genes acquired from these species, but you can see that these pectate lyase genes are closely related, just as this clade of species are closely related as well. Another interesting one to note is the leuconostocs down here. And over here we have Bacillus. On the pectate lyase 412 amino acid sequence gene tree, we have similar patterns as well. We have, oh, my bad. We have, right here, we have the mitchneri clade, and over here we have leuconostocs and whatnot. And so taking a look at the species tree of lactobacilli, we see similar relationships. Over here, we have Leuconostoc citrium. Over here, we have bacillus, and they're pretty closely related to other lactobacilli. And so it was interesting to have found this kind of pattern for the pectate lyase gene, and because of it, we wanted to see if there were any other quorum sensing genes that showed the same. And so that's where I looked into the histidine kinase and response regulator transcription factor gene. And so here we have the HAMP domain containing histidine kinase gene tree. And it turns out that this gene tree also showed evolutionary relationships similar to that of the species tree of lactobacilli. So the histidine kinase genes from the Mitchell clade are closely related to each other, just as the species are closely related to each other. So over here we have the Apolactobacillus bacillus Mitchell clade. We have Apolactobacillus mitchellii. The histidine kinase genes from Timberlakei, Canoei are also closely related. And then over here we have histidine kinase genes from Apolactobacillus kunckii. Over here we have from Apolactobacillus ozensis, Kimchikis, collonoidus, and paracollonoidus. And on the response regulator transcription factor gene, we found similar relationships. Again, over here, Mitchellii, Timberlakei. Over here we have kunckii. We have ozensis. Over here we have paracollonoidus, collonoidus. And so, and these are just like a few of the closely related species out of the many that have shown up on these gene trees. But when we actually take a look at a species tree of lactobacilli, we see similar patterns from these gene trees on the actual evolutionary relationships of the species. So over here, down here, we have the Apolactobacillus mitchellii, Timberlakei, and Canoei being closely related to Apolactobacillus kunckii. We have an ozensis right here. Over here, we have collonoidus, paracollonoidus, Kimchikis. And so basically, these gene trees, the relationships between these genes is the same as the relationships between the species. And so what exactly does this mean? Because the branching patterns in the gene genealogies are closely resembling the branching patterns found in the species tree, this is indicating to us that the Apolactobacillus mitchellii clade vertically inherited these quorum sensing genes. This is also further supported by the fact that genes from distantly related species weren't closely related to the Apolactobacilli genes in the genealogies. And then so for future directions, I'm actually currently working on carrying out enzyme assays for the pectate lyase gene to see if it's functional or not. But we also want to carry out these assays for the other genes as well. And if they are functional, we want to carry out further experimentation to find out what signal transduction pathways these genes are associated with, and to see if repressing the expression of these genes would have actually any significant effects on wild bee development. And so with that, I'd like to thank Dr. Quinn McFrederick and the Mark UCR program for their guidance and mentorship, the NIH for funding this project, and to the Undergraduate Research Symposium for inviting me to present my research. Thank you. And if there are any questions, I'm more than happy to answer them.